All right, guys. So today we're going to be talking about gel electrophoresis, specifically when we're running a gel electrophoresis with proteins. And when we do that, we usually run it with an SDS polyacrylamide gel, or SDS page for short. And so before we start, we really want to talk about the four biomolecules. And as we all know, there's four major ones. There's carbohydrates, lipids, proteins, and nucleic acids. And over the course of the next few weeks, we're going to be talking about both proteins and nucleic acids. But for today, we're just going to focus on proteins. And so, what are proteins? Obviously, as we discussed, they are a biomolecule, and they perform functions of all living organisms. Without them, we'd basically be nothing, because all we are is just a bunch of proteins put together. And so proteins are long chains of amino acids, or what we would call a monomer. So if you put a bunch of amino acids together, you have a protein. And so amino acids are the subunits, or the monomers. And so a long chain of amino acids, as I said, is called a protein, or if you want to be more general, a polypeptide. We have 21 amino acids, and amino acids can be charged or hydrophobic and or hydrophilic. So they can either be attracted to water or be repelled by water. And so, for example, if we look at myoglobin, myoglobin is a protein that basically binds oxygen and iron together, and it's found in the muscle tissues of vertebrates. And so obviously this is very important for our bodies, because our muscles need oxygen. And so if we're running a lot of blood through our muscles, and that blood, which has iron in it, is also attached to oxygen, then we're getting a lot of oxygen into our muscle tissues, which prevents cramping. And so, as I said, with a protein, we're usually run it through an SDS page gel. So we're going to kind of break that up. Let's first look at SDS. So SDS is sodium dodecyl sulfate, also called lauryl sulfate, and it is an anionic detergent. And so anionic means it's negative, and detergent means that it's basic. And so when you attach an anionic detergent to anything, or dissolve it in anything, it's going to have a net negative charge, which is going to be really important coming up later. And so if we coat our proteins in a negative charge, we know that a negative charge and a negative charge is going to repel each other. And so that means that we can break up some of that coiled structure of proteins. So if we go back and we look at myoglobin, right, we can see that myoglobin has all these little twisty kind of structures, right? It's very coiled up, a lot like DNA. And so if we want to run it through a gel, we're going to want to straighten that out. And so if we have a bunch of coiled protein and we just start adding negative charges everywhere, the negative charges are going to repel themselves, and we're going to have a straightened out structure instead. So that's step one. However, a lot of proteins have what we call disulfide bonds, and unfortunately our SDS doesn't do much to break up those bonds. And so what we're going to want to do, oh, and I'm sorry, this is an example, right? If this is our protein, we're going to surround it with negative, and so we're going to surround it with a negative charge, and so then our coiled structure, okay, is going to straighten out. And like I said, we still have those disulfide bonds that we need to start breaking up. Okay, and to do that, we use DDT, or I'm sorry for the pronunciation here, but dithiouretol, or DDT, which reduces disulfide bonds because it's a strong reducing agent. So remember that we went from a coiled we then basically added SDS, which made everything negative. And so now we have this straightened out structure, which a bunch of disulfide bonds. And so then we added DDT, which is a strong reducing agent. And instead, now we just have one straight protein, okay, which is ideal for running it through a gel. We also want to use heat. And so many proteins have hydrophobic regions and can be tightly associated with other molecules such as lipids, right? So if you have a hydrophobic region, which is afraid of water or repelled by water, it's going to be attracted to lipids because lipids separate from water. And so heating samples shake up the molecules, which allow SDS to bind into the hydrophobic regions. And so, you know, if you have a tightly coiled protein that is also hydrophobic, then it's even more tightly wound, right? And it's going to look something like this. And so we need something that's going to get into that hydrophobic region. And so if it's surrounded by lipids, which tend to be relatively solid or hard to get into, they have really tight structures, then by adding heat and adding kinetic energy, right, we add this heat, then all those molecules are going to shake up and it's going to become slightly looser. And when we have that slightly looser structure, it gives us an opportunity to then insert those negative charges in order to break it open. And so it allows for that final denaturization. So once we have broken up our protein, we're ready to set up our gel separation. And so polyacrylamide creates a matrix that molecules, in this case proteins, have to navigate through. So inside of this casing, okay, is a polyacrylamide gel, okay? And in that gel, there are a ton of little pores, okay? Think of it as almost like tunnels or a sponge, okay? So inside the gel, there's a bunch of little holes, okay? And so if this is the top where we insert our samples, okay? Well, our sample is going to have small pieces, large pieces, and medium pieces. And so if you think about all these obstacles, right? What you want to know is that the smaller molecules, like these little purple beads, okay, they're really small, so they're going to be able to navigate all these tunnels a lot faster than the larger gels. And so when we run our gel, we would expect them to travel farther, okay? And then our medium beads, okay, will probably travel pretty far, but not as far as our small ones. And then our large beads, okay, they're going to have a really hard time getting through all these pores and all these little tunnels in the quote-unquote sponge, so we wouldn't expect them to travel very far. So size matters, okay? It's a mixture of molecules. So, mixture of molecules, okay? But what makes them travel from one side to the other, right? Well, we actually run an electric current through the gel. And so the top where we put in our samples has a negative charge. And then the other side of the gel has a positive charge. And so if our sample goes up here, then we have a negative charge. And then at the bottom, we have a positive charge. Now you remember that we coated our proteins with SDS, right? So our proteins are covered in SDS. And remember that SDS is negative. And what we remember is that negative and negative repel each other. And so if they're repelling each other, these macromolecules are trying to get away from that negative charge. And they're going to move towards the positive charge because we know that opposites attract. Okay, so that's what's making the macromolecules run through the gel. And that's why we call it electrophoresis. Okay, we have the gel and then we have the electrical current. So the final separation is dependent on differences in relative molecular mass of proteins. And again, what we're talking about here is that small molecules are going to have smaller molecular masses, which means that they're going to travel farther because they're better at going through all those little tunnels and through all those little obstacles, okay? And then we know that large molecules have larger molecular masses and are therefore not going to travel as far. Okay? So we express mass in what we call Daltons. Okay? And remember that weight is not mass because weight is also known as a relative molecular mass, which equals to the ratio of mass to 1/12 the mass of a carbon-12 atom, okay? So just remember that we are measuring mass and not weight. So once we've run our gel, okay, this is an example of what a gel looks like, right? So this is the top where we would insert our sample. So this is the negatively charged area, and then this is the positively charged area. So as you can see, we've run our gel and we can see all these bands. Okay, these are all bands that we've staked. However, how are we supposed to tell how far a band has traveled? And even if we could tell how far it's traveled, how do we know its molecular mass? Well, we can determine relative molecular mass, right? To determine the relative abundance of major proteins in a sample compared between species using known proteins, okay? So this is really important. When you run a gel, there's no way you're going to be able to tell the relative molecular mass without having some known values, okay? And we can, so known proteins. And so we call those known values either standards or markers, okay? These are commercially available proteins. They also have DNA or RNA with known size ranges, usually stained with dye so you can follow its progress through the gel. So when you're running your gel, right, you'll have, anywhere between 3 to 7 samples. And your samples actually won't show up, and you won't be able to see those bands until you dye them. However, when you're using something like a kaleidoscope standard, right? So again, it's that commercially available protein with known size ranges. They come pre-dyed. So when you run the gel, you can actually see the kaleidoscope, okay? And so, for example, the kaleidoscope standard that we're going to be using, okay, so the kaleidoscope standard that we're going to be using, right, is going to show us myosin, beta-galactosidase, BCA, carbonic anhydrase, soybean trypsin inhibitor, lysosome, and protein, okay? And so when you get the standard, it's going to come with a worksheet, and it's going to say, okay, when you see the first band, that's going to be myosin, and that's going to be about 210 kilodaltons. Okay? Same thing with actin, okay? So you know how much myosin is, and so after that, you can basically guess other bands that are on your sample. So if, for example, myosin is 210 kilodaltons, and you get a band up here, well, you can probably guess that it's going to be actin, okay, because actin is about 44 kilodaltons, okay? So you use this, your kaleidoscope standard, and your standards or markers in order to compare your sample values to known values. And from there, you can try and figure out what you're looking at. And this will all make more sense when we do the lab and when we look at an example sample. But for now, if it's a little confusing, it's okay. Just make sure that you're either asking questions of your instructor or you're waiting to see an example that will clarify this.